Hello friends, in this video we will explore the challenges in the single cell data. Okay, so not all the platform is perfect, so every platform has its own limitation, and the same applies to single cell technology as well. So the data what we get is not perfect, so there is lot many issues are there with the data. So in this video we will talk about them. But to give you a recap, in the last video, we talked about how the single cell technology works. So basically, we explored the cell capturing, how we isolate the cells, how do we capture them. We talked about different method by which we can capture them, and then we talked about like once we capture them, how we prepare the molecule. That means attaching the cell barcode, attaching the UMI. Then we explored the way we convert the fast queue to count. Basically we do cell demultiplexing, then UMI deduplication, and once we get the count data we explore like in the downstream analysis. So we explored how the single cell technology works. So now in this video we will start with understanding the different challenges we encounter in the single cell data. Okay, so first let's see what can go wrong. Okay, so in the ideal situation, in a particular droplet, one cell should be captured. Okay, so ideally there should be one healthy cell that to be okay. It's the cell should be healthy and that should be captured in a cell. So that is the ideal scenario. But sometimes what happens, we get empties. So basically, instead of capturing cell, uh, you the drops that in the drop, some floating RNA or ambient RNA will be captured. So now you can. So what is an ambient RNA? So when we do the dissociation, right? So in the first step, when we break the tissue to cell, sometimes what happens, cell dies or not dies, cell membrane breaks. So what happens, the cytoplasmic RNA gets leaked out from the cell membrane, and those will be freely roaming around, okay, in the cell suspension. So that means they do not belong inside the cell, they're outside the cell now. Okay, so the cell is broken and all the RNA got out. So such empty RNA by it will be captured in the drop. So this is an issue. Okay, so ideally you will not find cell, instead of that you got empties or ambient RNA. So generally we call them empty. And another case will be there sometimes, instead of one cell you capture two cell or doublet or multiple cells got captured in the droplet. Okay, so that is one issue. So what will happen, you will overcount, means that the gene. So basically when you do the sequencing, okay, the genes from this and this everything you will be count. Okay, so that's where it's an issue. And the third thing is like you capture a cell but it is not healthy, it is actually either broken or it is dead. Okay, so dead cell got captured. So what will happen? Okay, so let's see now how we will handle this situation. So in the first case where you got you empties, so basically you did not capture cell, instead you captured ambient RNA. So in that case what will happen when you do the sequence it, these mRNA will be get sequenced, and when you do the what is that, when you do the cell counting from this cell, only few genes will be mapped, because hardly, if this cell would have captured a healthy cell, ideally a healthy cell, let's say a cell to in order to survive, let's say hypothetically 5,000 genes minimum would be required. So now, but since no cell is there, if you count how many genes are expressed from this cell, let's say you got 100. So now with this you know that, oh okay, at least 5,000 genes should be expressed, then probably it's, I can at least tell it is in cell, because in order to function a cell we need at least this many genes. But in this drop I found that there are hardly 100 genes. So maybe this is not a cell, instead it has captured the empty RNA. So using this criteria, basically based on small number of genes expressed, so we filter out. So any cell where the number of genes expressed is very less, so we you discard them. So because they are not cell, because ideally for a cell some amount of genes should be there. That logic we apply. Okay, So the same opposite logic means where in case of doublet, what will happen? More genes will be expressed because these genes and these genes from both the cells will be, both the RNA you will be sequencing, right? So now the amount of RNA is more, so you will get more reads. So that way we can filter out if a cell is expressing very higher number of genes, then we suspect that maybe that could be doublet. So if a gene, if a cell is cell is, has less number of genes, so we guess that okay, it is empty. If it is higher range, very high, then you say that it is a doublet. So in our Surat pipeline or any tool you use this cutoff, like remove cells where there are less number of genes and even higher number of genes, you exclude them. The third category is the broken or dead cell. So what happens when a cell dies? So what happens, and why cell dies? Because you are giving lot of stress, right? When you are breaking the cell through enzymatic digestion and mechanical digestion, the cell goes through lot of stress, and because of that, cell dies. And when cell dies, what happens? There are two things. When a cell dies, apoptosis gets triggered, and the enzymes degrade all the cytoplasmic RNA, so that uh, but the mitochondrial RNA will remain intact because mitochondria has its own membrane. So those will get protected from the enzymes of cell degrade cell cycle, sorry, the apoptosis kind of thing. So that way, when you sequence this, part, so you will see, mitochondrial, the reads will be aligned to the mitochondrial gene. So you can, using that criteria, any cell or any cell, means any droplet from where a majority of the genes are aligned to the mitochondrial genes, so then you can guess that that is definitely a broken cell or it's a dead cell. So with that logic, another another contribution what we can do is like, how many are aligned to the ribosomal content as well. Higher ribosomal RNA is also there, then also you can discard them. So these are the criteria by which we remove empties, doublet or multiplet, even broken and dead cell. But apart from that, there are some different other issues are also there. The first number is generally, first issue is another called dropouts. Okay, this is called gene dropout. So what happens, there are two. I'm just explaining here. So two possible reason for a zero in the single cell data. First is real zero, means ideally that gene is not expressed in the cell. So that means no RNA and no detection. So that means this is good. This is the real zero. Means ideally this is, we know that gene is not expressed at all. But sometimes we get artificial zero or gene dropout. Means gene is expressed, but your technology could not detect it. That is the limitation of the platform itself. The 10x genomics is not good enough to detect it. That is the error of that technology. But you can't do anything because the technology is doing the wrong thing, right? You cannot do anything. So but we have to deal with it with the data. So that's where in the single cell data you cannot differentiate whether that zero is actual zero or it is artificial zero, or this gene dropout. Ideally all the zeros should be real zero, nothing should be in the the gene dropout artificial zero. So that should be our objective. So yeah, that is one issue, like higher number of dropouts, that means a gene is expressed but the expression is not detected due to the technological limitations. Okay. And the second issue is like data is very noisy. Second challenges with the single cell data is the data is very challenging. Sorry, data is very noisy. So what does it mean? High level of variation between cell to cell. See, although you sequence, since we studies each cell separately, right? Problem is like each cell is very different from each other. So now how will you analyze them, right? So why the cell to cell you will see lot of variation. First is like capture efficiency, means each cell you are capturing mRNA, right? So each cell you are sequencing separately and you are breaking that cell, isolating mRNA and attaching the barcode, attaching the UMI, and you are sequencing them. So that capture, how much capture efficiency, that will vary from cell to cell because each cell is considered as in a different experiment, consider. And that's where you see some technical variation between cell to cell. Okay, the second thing like reverse transcription again. So once you have the RNA to DNA conversion, right? So that requires the enzyme, and that enzyme you are adding into each reaction chamber separately. So that means parallelly so many reactions are going on, and each cell will be different. That the transcription, reverse transcription efficiency will be different from each droplet to other droplet. So in that case, what will happen? You will see some variations. Some RNA will not be converted to cDNA. So that means you could not detect them. So that way, this is another technological variations. We get. Third is amplification bias, means you are doing PCR. So what will happen? The PCR is, in some droplets, PCR will be very good. Perfect amplification happens. But in some cell, maybe reagent is not sufficient, or maybe some technological limitation is there. Because of that, PCR did not happen very well. So in that case, what will happen? You could not detect some of the RNA molecule. So because of that amplification bias from cell to cell, you get technical variation you will see. Also, sequence significant difference in the sequencing depth. Number of UMIs per cell. In some cells, you will have you will sequence in very higher depth, but in some cell the sequencing depth will be low. That variation will also exist in our single cell sequencing data. And last and very very important one is the cell size and cell cycle stage. So right, we have cell cycle, right? So a cell go through different cell cycle stages. Like we have G1 phase, the first growth phase, then we have the G2 phase, the second growth phase, then we have the S phase, where now cell is already grown. Now it is in the preparation to the cell division, because mitosis, right? Cell has to divide, it has to produce its offspring. Okay, so it will now it enters to the S phase, where the cell division will start, and finally the M phase happens. So now these four stages, the is very different. Cell is very different, right? So the genes or the RNA, what you see, if a cell is in G1 phase, will be different from G2, from S and M. So now the time when you captured the cells from the tissue, it might happen that some cells will be in their G1 stage, and because those are newly formed, because recently they are born. But some cell will be in the G2 phase, some cell will be in the S phase, and some cell will be in the M phase. So ideally, the cell to cell, when you compare, that variations you will see. So ideally, when we do the analysis, we should exclude this technical variation. Ideally, we will do regress out. We'll talk about that in pseudo relation. But we have to use some statistical method by which we will have to ignore, or remove that cell cycle variation from the data, because we are not interested in the cell to cell subtype because of the stage. We are interested in actually how biologically we will see, let's say fibroblast or other different subtypes are there in the given tissue. So that's where data is very very noisy, and you will see high level of variation between cell to cell. So that's where the data analysis challenges comes. You have to do normalization, you have to do scaling, you have to do transformation, lot many things you have to do. You have to do filtering, and you have to find various genes, highly variable genes. Lot many things we have to do because of these regions. Okay, these regions. And yeah, next is like complex distribution of expression value. And expression value, the range is very different. Means some genes, the expression value be in the very lower range, let's say 10, 20, 30, right, read count. And for some genes it will be in the 1,000, 10,000 range. So you will see a very high variation in the distribution pattern. And that's why whatever the method has been developed for bulk RNA sequencing cannot be applied or don't work for single cell RNA sequencing. Okay, yeah, that's where the analysis is actually challenging. We talked about that. And another thing, like batch effect might come. For example, when you're doing analysis, if you have different batches, you have done the experiment, then you have to also see whether there is some batch effect there in your data or not. If yes, then we have to do that batch effect correction as well. So we talked about cell cycle stages correction. We need to correct this cell cycle variation. Again, we cannot differentiate the real zero versus artificial zero. Yeah, so I think that's all about in this video, where we talked about different challenges we see in the single cell sequencing data. And yes, in the next video we will start with the Cell Ranger. Basically, this is the tool provided by 10x Genomics, where we can convert the raw the output from the 10x Genomics to ultimately we'll do the counting. So we'll talk about the Cell Ranger in the next video. I think this video was useful to you. If yes, I hope this video was useful to you. If yes, then please like it, share among your network, and subscribe to my channel if you have not yet. And thanks a lot. So we'll see you in the next video for the Cell Ranger. Yeah, bye bye. Goodbye.